This is a production of Cornell University.
Thank you, everybody. I'm stunned by the turnout here, and I hope I will do your expectations justice. When Angela got in contact with me, she asked for something that would stimulate discussion, and obviously would have something to do with soils and carbon cycling. So the general thrust that I chose is this old thing that we all learn at the very beginning of our scientific careers. This be careful with correlations. I think that is this mantra that is very early in our scientific education as maybe the data look good, but always be mindful that you need to have some sort of underlying explanation. And I thought I use this as the framework for what I actually want to get across. So I don't want to tell you that you need to be careful with correlations. It may look like I was going to do that over the next, let me say, 5 or 6 slides, but don't fall into that trap. So what do I want to talk about? First of all, the talk about carbon cycling is replete with the use of this word, control. And it pays to think for a moment what a control is. So when you want to know what a word means, you go to the dictionary, don't you? And when you do that, then you find that a control is defined as the power to make decisions, or the ability to direct actions, or an action itself, or a method, a law that limits the amount or growth of something. So a control is perceived as something that is actively doing something. When we accept this choice of definitions, then we are immediately aware of the relatively complicated nature of this thing, control. When we have actions, decisions, methods, laws, abilities, then we have to accept that this entity, this concept of control is variable, dynamic, adaptive, structured, multifaceted. I could go on and if I put a little effort into this. So what I'm trying to get across here is controls are not single numeric values. One number is not a control. Or even a sequence of numbers is not a control. And even simple mathematical functions or algorithms do not qualify as controls. Always when you follow that Merriam-Webster definition. So now let's try to find the connection back to carbon cycling. When people talk about carbon cycling and when they model carbon cycling, then they often use controls with the interesting development that they multiply over the years, or the controls that are being talked about increase in number. We can broadly distinguish environmental controls, which include drivers such as moisture, precipitation, radiation. Heat is a form of radiation, but there's also visible light, ultraviolet light, high energy radiation. It might be ionizing. Structure of the environment can be parameterized as total pore space, pore size distribution, preferential flow paths, mineral surface chemistry, resources. So all these are environmental factors that are known to have an influence on the cycling of carbon. Oxygen availability. You take the oxygen away and things slow down dramatically. Alternate electron acceptors. You take the oxygen away, but you provide nitrate. And things keep going, at least for a larger part of the microbial community. Proton concentration, other word for pH. Catalytic metals, organic co-metabolites that will return mineral-borne nutrients. So something in the order of 11 or 12 environmental controls. Substrate controls. So this refers to the fact that the object of decomposition has certain properties which tend to be of relevance for the decomposition process. So which are these properties? Bioenergetic potential. How much energy is stored in that organic molecule? How attractive is it from an energy viewpoint to the decomposer? Oxidation state of the carbon mainly. This is an important variable that determines how difficult it is to get at that energy. But then also we are not just decomposing things for energy. Not us and not the microbes. We sometimes need nutrients. And in that case, the stoichiometry of the nutrients is important. And this not just for the big ones, the big mass nutrients, but also for a plethora of micronutrients. Assimilability. So your compound may be loaded with energy, contain tons of nutrients, but it may have some other feature that prevents it from being assimilated. One really important number here is molecular size. When we go above 600 Dalton, we get into real problems getting this compound through the cell wall, from the outside to the inside. And the way these things are done in nature means that thread-like molecules are much easier to take in. So, but not everything's a thread. And this is what we mean with assimilability. And parametric expressions of assimilability can be aqueous solubility, molecular size, linearity, globular structure. All these features influence control, organic matter turnover, accessibility. Where is it? Is it far removed from the decomposer, or is it close by? Is it co-precipitated with a mineral phase, kind of mired in concrete? Is it absorbed, encapsulated in an aggregate? And that's another dozen or so individual controls that we know have the power to influence the turnover of carbon. So, and I'm not done yet. Organismic, so the decomposers, those who are actually doing the business, they have different lifestyles. Grazers, predators, mycorrhizal, free-living fungi, bacteria, archaea. Microbial diversity, who is there? Who is dominating the microbial community? How many of them are there? How active are they? Are they all dormant, or are they competing with each other massively? Are they synergistic? Are they cheaters? There is such a thing. Those who lie in wait for others to secrete all the expensive enzymes. And growth stage. Physiology of microbes, and especially the chemistry of the extracellular polymeric substances that they secrete, change as a function of growth stage within hours. Ability to use oxygen, that's the second most electronegative element in the periodic table, and it's the strongest electron catcher. When you have a system that contains oxygen, you can make these large energy gradients that really drive things, and when you cannot take advantage of this and your energy gradient is only like that, such as between your substrate and iron, for example, then the energy that you can harvest is much smaller. So if you really want to have things turn over quickly, you want to have oxygen. And that is reflected in the biology of the decomposers. So some of them are really specialized on this, that others, once in a while we run out of oxygen. And then you have those who are absolutely unable to deal with oxygen, organisms that probably are somewhat left over from the days 2 or 3 billion years ago when there was no oxygen, free oxygen. But then there's all kinds of intergrades here. So what I have here is something in the order of 30 or if we were to engage in a collective brainstorm, we would be able to come up with another 10 or 20 within 15 minutes, I'm pretty sure. So the challenge then is how do we constrain this? What matters? Who or what controls the control? Is there a hierarchy of relevance? Do they depend on each other? Is the control number 7 of the same importance in Cornell as it is in Corvallis? And in the the respective soils, of course. So how have people dealt with this? I mean, we are not the first who realize this multitude of possibilities. So others before us have realized the same thing, and they have developed strategies how to deal with this. And one of the strategies was the idea to look for a master control for one parameter or for one item that controls it all, the one ring. And that one ring, the first big popular one ring, is what we call substrate quality. That's the first suggestion of a useful master control that was extensively tested by the scientific community. So I'm presenting two examples for the application of this principle of substrate quality in the function of a master control. You probably have all at some point heard something about humic substances, and there is a paradigm, a still standing paradigm, that these humic substances tend to have old radiocarbon age because there is a process that is called humification which makes them highly aromatic, and this then explains their resistance to decomposition, which explains the high radiocarbon age. And this goes back to a benchmark paper published in 1967. So it needs a little explanation. So you have a graph with age on this axis. High age is here, 1500 years. The numbers are a little bit small, but so old carbon is here, and this is 1500 years, 1500 years. And then you have an index for aromaticity. This is the ratio of the light extinction at 400 and 600 nanometers. This is a way of determining how many aromatic compounds are in that specific solution. So when you are high on this axis, the aromaticity is low, and when you are high on this axis, the carbon is old. So the young carbon, or the young age, is following this red line, and that age gets older as the aromaticity goes down. Increases. I'm sorry. The brown line goes down, but that means the aromaticity increases. I was practicing this for a couple of times, and I was afraid I get it. So at this point, I want to come back to my little introductory remark about the importance of correlation and our joint love for correlations. We all, when we see something correlate like this, the age goes up when aromaticity goes up, then we all love this. We cannot avoid that. We really like this type of observation. And what we do then is we inadvertently, instantly invent a model, so some sort of explanation. And in this case, the explanation was really plausible because we know that aromatic rings require a lot of activation energy to break them. So it makes total sense for these two parameters, age and aromaticity, to correlate. But in order to demonstrate that they do indeed causally generate this resistance to decomposition, one would have to demonstrate that. And actually went one sentence ahead of myself here. I was going to mention that these ages here, this is what we in carbon cycling models consider the passive pool. You probably have heard about the active, the intermediate, or slow, and then the passive pool. So this is what gave rise to this passive pool idea. 1,500 years of turnover time. So what are the issues with causality here? This was an accepted relationship, and it was is still an accepted model. This type of inference informs the community land model, which is supported by NSF and DOE, and which is the basis of climate modeling at this time as we speak. So what is the issue with causality? So I'm saying that the ideas behind the correlations used in the existing CLM are not based on causality. So decomposer organisms have no way to sense how much 14C or 13C is in a glucose or in a lipid. They cannot make a selection based on 14C content or 13C content. They recognize the glucose or they recognize the lipid, and then they decide whether they want to engage. So old age is not an argument for stability. It were an argument for stability if the decomposer would have a mechanism to select for young versus old, which they don't. They go for chemical characteristic and not for radiocarbon content or 13C content. Fungi are absolutely capable of decomposing condensed aromatic rings. There's a huge literature, and I just did not put it up. You just Google PAHs and fungi, and you get 10,000 hits or something. So the mechanistic assumptions behind that little logical chain that I had on the previous slide are not corroborated. The other thing, or the third major argument here, is the early researchers were working in Canadian grassland soils. And they had an important piece of information. They did not have that. We have it today. So they see this 14C age is a function of aromaticity, and they don't know an important feature of these grassland soils that we know today. And what is that feature? The aromaticity in these soils does not come from whatever that humidification process represents. It was supposed to be here, the map. So you have these grassland soils, which are zonal soils. They are all along somewhere between the 20th and the 50th parallel. Actually, a few down here in Argentina as well. This is a map using the FAO soil systematic, so it has the phaeosemes, chernosemes, castanosemes. These soils are broadly equivalent to the soil taxonomy molly soils. These grasslands in their native state, they burn regularly, and that loads them up with black carbon. BC means black carbon. So here's the data. So the study is based on 28 soil profiles, very rigorously investigated with radiocarbon and 13C markers for black carbon, and you find that there is something of, the line was not drawn here, but there is something of a correlation between the black carbon in grassland soils and the carbon stock in these soils. And the takeaway message here is that grassland soils contain between 1 and 5 kilograms of black carbon, which is more or less a concentration of aromatic rings. So we are facing a long-standing paradigm, and there is abundant evidence that the mechanistic underpinnings are no longer valid. Let me put it that way. Oh yeah, and this was published by Rodionov in 2010. So let's leave the slow pool, the 1,000-year pool, behind for a moment. There's another example of this attempt to use a master control to describe carbon turnover. And that is the paradigm that litter decomposition rate should be a function of the lignin-nitrogen ratio. This is an absolute citation classic here, Melillo et al. 1982. And this was the work where it was shown for the first time that the biomass remaining after you expose litter for a certain amount of time, or let it decompose, the amount of litter that remains after time x is a function of lignin-nitrogen ratio. Basically, you have high nitrogen, the litter is decomposed quickly, and you have low nitrogen, or the inverse is little lignin, high lignin, then the litter goes away much slower. So, which leads to this this general paradigmatic statement that litter decomposition depends on lignin content. There is one caution that one should immediately become aware of when one looks at this graph. So, if this is a categorical predictor, then the green and the blue should be on top of each other. But apparently, this relationship can be modified. And here, we run into this issue with our assumption that a master control may help us to get a handle on the multitude of controls. If, for whatever reason, I want to be able to distinguish between ecosystems, apparently, just one control is not going to carry the day. So this is, by the way, the experimental evidence that gave rise to this idea that we should have a slow pool in carbon cycle models. So because this type of material turns over on an annual, multi-annual time scale, 10, 20. The issue with this is not only that the green and the blue were not on top of each other. The issue is also that this old assumption, lignin is only decomposed after the labile or N-containing organics have been used up. So this is this Berg and Staff 1980, this litter decomposition model, that you have the mass of the litter, the blue line, mass on this axis, time on this axis. As time progresses, the litter comes down, the mass comes down, and the underlying idea verbalized here was that the lignin doesn't respond until the labile stuff, which would be the area under this curve here, until that is decomposed. So the idea, the bugs or the decomposers go for the easy stuff first, and when they have used that up, then they eat basically they eat the candy, and then they go for the potatoes. So again, this is somewhat plausible because it is in line with some of our own behavior. Just was talking about the candy and the yeah. No, this is we have evidence that processes of this kind can work in this fashion. But is it actually what is going on here? And then there is new research that shows vigorous lignin decomposition is going on only as long as there are these easily decomposable things. So the mass loss, now again, the litter mass loss, the whole litter mass loss, described by the blue line, but the lignin gets decomposed here at the front. When you have these things in order to help decompose the lignin. But when you run out of the accessory, the so-called co-metabolites, then the resources to do business with the lignin are no longer there, and then the line flattens out. Published in 2011. So, and the old model, 1980. So it's 30 years, and new evidence has come around. In both cases, it's not my intention to criticize our predecessors, but my intention is rather to point at the need to be careful with these correlations and don't trust them too much because if we do, we might miss the things that are really going on. So yeah, and I was going to point that out, which I believe I did. So is substrate quality an appropriate master control? Well, based on the evidence I have just presented, I hope you can join me in saying that is probably not so. And if that is not so, then is there maybe some other master control? And here we come to work that was published in 1981. So large differences between the turnover rate of particular compounds in liquid, microbial cultures, and in soil indicate that soil provides a measurable degree of protection. So those who set out to get a handle on the controls focused on the substrate. And but then somebody realized the soil controls, the matrix of the soil controls decomposition as well. So you may have the substrate somewhere here, but then it matters what kind of matrix this substrate interacts with, if at all. Interestingly, look at that date. That kind of thinking came around more or less at the same time. But the substrate control idea was way more appealing to the scientific community. Liked it much better. Didn't really want to deal with this matrix and mineral stuff for a while at least. So until 1997, Margaret, my former boss, was lucky with a Nature paper that was titled Mineral Control of Soil Organic Carbon Storage and Turnover. So this then is an example for an attempt to address some of the shortcomings that the substrate quality idea had by replacing it with a better master control. The problem with that approach was that it's not exactly a straightforward predictor either. So, and it would be so if you take mineral components from 3 different soils, here this is the clay fraction, then you have to accept that the ability of clays to associate with carbon is much different between the soils. So you can't just say, which actually the Century model does, I take clay content and use that as a rate modifier. It matters a lot what kind of clay you have in there. So what do we make out of this? Where do we go from here? A contest of the master controls does not seem to be the best strategy here. So remember in the beginning I took some time to show that there is no problem to come up with at least 30 or 40 individual factors that would qualify as some sort of control. So picking one and focusing on one of these controls is apparently not the best strategy. Why? Because in both cases the evidence in support for the importance of that control was largely correlative. The researchers saw this correlation, the lignin-nitrogen ratio and the amount of litter left. Perfect, straight line. You go out, do some experiments out there, and you have 10 data points, and you get a straight line. How often does that happen? It must mean something, doesn't it? Yeah. But the fascination with this type of result then makes us so confident that we do not dig deeper. So are we doing enough to identify and understand mechanisms and causes behind the assumptions that we use to inform carbon cycle models? I'm positing here we are not, and you are the generation that has to change that. Brings up the issue of causality. So how do we get at causality in carbon cycling? What are ways to make this work? So that controls actually are descriptors. The thing that we need to understand, we don't need another label we already have 30 or 40. What we need is understanding of the system, the machine, the engine, reciprocating or turbine. That type of understanding is what we need. And a good way to approach this, or the way that I am proposing here, is to look for where the soil comes from, evolutionary adaptations. So the number, relevance, connectivity of controls, our catalog of 30 controls, can they be seen as a consequence of systems design? Which would have, if this in any way works out, it would mean if we get an idea of the principles behind systems design, how is the soil as a place where carbon is processed, how is that designed, how is it laid out, what are the guiding principles? If we know that, we may be able to get at the causalities that we are missing in these old correlations. So what I'm saying here is in disagreement with the views that geologists have about soils. Geologists take soils as the interface between a planetary body and what is beyond. And they do not require life to be involved in the formation of soil. This understanding of soil, when I say the soil evolves with the ecosystem, goes back to this idea of that Berner, I'm sure others as well, have put forward, that when soil formation really took off on this planet, when plants started to evolve, when the algae came out of the water and inhabited the land, that is the moment where we should start talking about soils. So if we follow that premise, and again, the geology community, folks who like to find soils on Mars, they have to do without trees and without roots, and so that's a different view of things. Evolution has created flow-through reactor systems with unique reactor design and carbon processing strategies. So that would be the key to causality. Reactor design, carbon processing strategies. So something like 400 million years ago, a few algae started to inhabit the land surface. These little algae evolved into protoplants, which developed proto-roots, which, by the way, from the first day were working together with mycorrhizal fungi and with bacteria, because they were there first. And so, as a result, the matrix gets transformed. And when you allow the plants to become more complicated and more powerful as biogeochemical machines, then you get these systems, and it is relatively easy to envision that when you change the ecosystem here, that would have effects on how the soil is functioning. You have different inputs, you have different quantities of inputs, you probably have other biota, and so you get a different type of reactor design and different carbon processing strategies. That now means the controls are the result of how the system works. You don't approach the problem by asking what does temperature do to the system. You approach the problem by saying, how does the system work? Oh, if I raise the temperature to 50 degrees, that means such and such. So it's a reversal of the line of thinking. The basis for this is the fact that within any ecosystem there's coevolution of above ground and below ground biota. The decomposers are there because of the plants. That's the idea behind this. And it's not my idea. It's Eastwood et al. It's an interesting paper. The science paper is only 4 or 5 pages, but the supplementary information are, I believe, 70. So you really... So this is... If soils evolve to decompose organic carbon at the rates and in the sequences required to support the ecosystem, that puts into question ideas like, oh, I have a certain lignocellulose that should decompose at a rate that is typical for lignocellulose. That's what our models do right now. 76 days, lignocellulose. If the system has evolved to deal with lignocellulose in a certain way, then the system determines, the characteristics of the system determine how long this lignocellulose takes to be decomposed. So it's no longer an inherent property of the lignocellulose. So system evolution, here is a dune in beautiful Oregon. And this is grad student Adrian Gallo taking a picture. And only about 500 meters away from this dune is an older dune. And you see how that soil has evolved. There's shown some E horizon, a BS horizon. So that's an incipient sporozoal. This is a very early antisol. And in the course of this development, there's change of the mineral matrix. It's consumed. Minerals are destroyed. New minerals are formed. And things are rearranged in the soil. Structure is changed. So the reactor design, this is a different reactor than this. It does different things. And those things that are happening different here influence the turnover time of the organic matter. Oops. Yeah. This whole matrix issue must be seen not as a static entity, but as a dynamic, adaptive part of the ecosystem. So the mineral matrix is actually shaped by the biota, which is the major force that makes the soils function in different ways. So a very crude idea of how this works is you need water. We all agree with this. Water comes slightly acidic because you have CO2 in the air, which dissolves in the water, and that makes it mild acid. This then allows for weathering of the minerals. You can have small reactive weathering products, and then they can be mineral partners of mineral organic associations. And the intensity along this path varies what you get here. Moisture also allows things to grow, microbes and plants. And then when plants grow, for example, they generate root exudates, which are acidic, really acidic. And that then augments or amplifies processes down here. So you have more growing here, you have more change here. You get the idea. You make this a thriving active part. You get I have the bad word here, humus, because it's in every book. This can be decomposed, and you again making something acidic, and this again then influences the properties and the shape of the mineral matrix. So a highly dynamic system. You take vegetation out, you change the intensity of processes, you mess with the microorganisms, you change the intensity of processes, and it should illustrate the contention that this is not very well predicted or described by saying lignin or cellulose are decomposed within 50 days. So, yeah, I changed the importance of any of these arrows, and that number will not have any connection with reality. So where are we? Current research practice is centered on understanding controls and their interactions, which can be paraphrased as understand the control to understand the system. That is how we all have been brought up. This is how our modelers work. They want us to figure out how a algorithm that describes how the control equation influences turnover time. But would it not be better if we accept that controls in their relative importance evolve with a system, to put more effort into understanding systems design, and such in a way that we can derive the relative importance of controls for the governance of the system. That would turn the thing around. Understand the system in order to understand the importance of the control. So if you are interested in temperature sensitivity of organic matter decay, well, you need to understand how decay works, and then you will be able to understand what happens when you manipulate the temperature. The way we are approaching this right now is from the other end. Systems design. So what does that mean? Soils are different. So like I just said, in Corvallis we have different soils than you guys have here. So there is an obvious difference between soil type. But even within one soil, you can identify regions where the process regime is completely different. So let's look at the litter layer here. This is a loose pile of substrate. So like walking into Starbucks in the morning. So everything's there, you just grab it. Well aerated, oxygen, plenty. No need to come up with any fancy physiology or so. Easily accessible to mobile decomposers. You have a force that we tend to forget very frequently, mesofauna. Those are the ones that chew the large pieces down to the size where the bugs can then do business. So they are abundant here. They can crawl among the leaves and twigs, and they are fast. They are usually the first ones on the scene. Susceptible to perturbations and to fluctuations of driver. A tree falls down, crashes on the forest floor, all this little paradise is annihilated. Or it doesn't rain for 3 days. The moisture holding capacity is basically not existing. So it dries out. The amplitude, ecological amplitudes are large. You need adaptations for that, basically. Let's go a little further down. A horizon. This is actually the region where you can say the ecosystem does its recycling. So this is the recycling facility of the ecosystem. You have an active input of co-metabolites through root exudates. So the plants, they have no interest. I mean, the plant cannot think, and an ecosystem does not evolve in a direction chosen by the ecosystem. It's all adaptive. So you excuse my language when I say the plants can support business by putting exudates out. There are regulatory mechanisms that allow them to do that in an adaptive fashion. The point is that as long as you have these plant roots, fine roots, you can really speed up things. You have something that is absorbed and protected, or it's a large chunk. You can support decomposition processes by meta inputs. You have a wide choice of catabolic pathways. Everything's going here. You have access to catalytic metals and reactive surfaces because you're already in the soil. You don't have that in the same extent here in the litter layer because you don't have the metals and you don't have the minerals. And the oscillations in oxygen supply are somewhat dampened because the moisture holding capacity here is much higher than here. But it's still cycling. Once in a while, you go through a redox cycle, and you can recycle those redox-sensitive metals that you can utilize as catalysts. Only when you get down here, then you are in the desert. But that is, again, that qualifies as a completely different operating condition for a reactor. Low substrate concentrations. A bit of substrate here, a bit here, a bit here, a bit here, and the microbe here in the center. How is this supposed to work? You cannot do very quick organic matter turnover under such conditions. Few organisms, lack of co-metabolites because no roots anymore. Reduced pore space because often you have clay formation at those depths, and that will clog your pores. It's difficult to get oxygen down there. And you have the issues of accessibility that I just pointed out. So this is what I mean with understanding how the system works. And then ask yourself, now when I am, let's say I am in this desert in the subsoil, and I put a chunk of wood down there. Is it really appropriate to say this turns over in 76 days as a substrate-specific parameter? I don't think so. So, in our zeal to understand controls on carbon cycling are relying too much on correlative evidence. Progress may come from bottom-up approaches that seek to identify the causality. That I'm giving you here what you already heard in your first statistics class. So what I really want to emphasize is it's worth to accept the fact that soils do not just look different, but also work different, especially when it comes to carbon cycling. The next generation of models and the next generation of scientists needs to emphasize the causalities behind soil processes. Don't do the same thing that our fathers have done. Now, when you see that straight line, you say, okay, I have a straight line, great. That is certainly going to be a paper. But be honest to yourself. Yeah, be honest to yourself. Do I really understand what's going on here? Have I tested whether my assumption is really the mechanism behind this? Or am I going the way of cheap sales? And if you're ever in a position to support some sort of modeling exercise, be especially careful with the causality thing. Yeah, a golden sentence to conclude. And paradigms must be continuously challenged and checked for compliance with causality. Doesn't that read beautiful? Okay. Thank you for your attention. Well, I need to admit that I'm not a modeler, and I also need to admit that I have enormous confidence in the possibility of technological advances. So I remember that when I entered the field 20 years ago, model exercises were done on supercomputers. Today, you don't need them. One of the major constraints was storage space at that time, and I understand these constraints have gone down. But I also remember that the computational complexity of models was an issue. And so, not knowing what the most recent developments in this field are, I think the argument that the models need to be simple, which I completely understand, and I think there is no reason to try to over-explain things. We know this from other phenomena. When you can explain something using 3 variables, then there is no need to throw 30 variables into the picture. In fact, principal component analysis is a tool that we all often use to bring down the number of variables, because it's then easier to make sense of what we see. So, my attempt to talk around your question is now going on for 1.5 minutes. But what you get from this is, I think there is probably some slack in what on the computational side can be done. So I think we should not be afraid to add parameters or add algorithms to our modeling attempts. But to what extent that can be realized is a little bit beyond my area of expertise. That okay, thank you. So I have friends. I have modeler friends, and they have played with not just decomposing organic matter, but breaking organic matter down into certain functional categories. And that seemed to be a useful approach that has been published last year, I think, in Geoscientific Model Development, obscure journal. Yeah. So if you are interested in detail, I'm happy to share the source. Yeah.
Thank you, everyone. Let's thank Markus again.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.